and welcome back to the World of Sharks podcast, the official podcast of the Save Our Seas Foundation. As the name suggests, this is a show all about sharks, rays, and the oceans. I'm your host, Isla, a scientist, diver, and all-round shark enthusiast, and every episode I have the very nice job of sitting down with experts in shark science and conservation to go on a deep dive into a different part of the wonderful world of sharks. This week we are diving into the chilly waters of my home country, Scotland, to meet an iconic cold water species, the spurdog. Also known as a spiny dogfish, a spurdog is a relatively small but highly mobile species of shark that is found in temperate seas across the globe and is common all around the British Isles. But while they might be small and relatively common, they are a fascinating species, not least because they are one of the few venomous species of shark. Yep, they are named after the two spines that you can find at the base of the dorsal fin, which actually secretes a venomous substance when threatened that causes a mild yet uncomfortable reaction in humans. Mind you, this only happens if you grab onto the shark, which I hope none of you would do. And also, they are typically a deep water species, only coming up into shallower waters to breed and feed. They also have a habit of aggregating in large numbers, and this is something that my guest today is very interested in. PhD researcher and Save Our Seas project leader Fenella Wood is a marine biologist and molecular biologist who is trying to understand why these groups form. And more specifically, she is using population genetics to figure out if the individuals in these aggregations are related. I'll let her explain why this is important and what it could mean, but this is just a fascinating episode about the curious world of the spurdog and a window into the equally fascinating world of shark genetics. It is just mind-boggling the amount of stuff that we can find out from a shark's DNA, and I loved learning from Fenella all about it. We also get into her career. She's looked at all kinds of things from diet studies to eDNA. She'll explain what that is in the episode. And now population genetics, and has worked with many different species from seabirds to angel sharks to now flapper skate, another very cool cold water species, and of course the spurdog. She's also worked at the Bimini Field Station in the Bahamas and on some really interesting projects. And I just think her research is so incredibly interesting and cool. And I think you will too. In this episode, we talk about Fenella's journey to becoming a molecular biologist and geneticist, get stuck into some spurdog facts, and learn what might be behind those aggregations. And we learn about concepts like relatedness and connectivity and why these are important to understand from a conservation perspective. So get your cold water gear together and get ready to head to Scotland. Let's dive into our episode. Hello, Fenella Wood, and welcome to the World of Sharks podcast.
Hello, thank you for having me.
No, it's absolutely our pleasure. I'm very excited today because we're going to be talking about one of my favourite species of shark, one which I've dived with a couple of times actually. Normally the sharks that we talk about on this podcast, I've not dived with or I've not seen that much in real life. The spurdog I am very familiar with, so I am so excited for this episode. And we're going to get into some cool genetics as well, some cool sort of molecular biology, which is, as we've just discussed, a field that I'm pretty much coming into brand new. When I did marine science at university, it was quite a long time ago, so I'm very much looking forward to getting into that. But before we do, we like to get to know you a little bit. Familiar listeners will know we like to start the podcast and end the podcast with the same question to every guest. And our first question is, what is your most memorable experience in the ocean?
So that's quite a tough one because I've been quite lucky and I love being in the water. So I've had some really amazing experiences. But one that really jumped out to me. And it's not particularly actually a shark experience, but it's slightly shark adjacent.
That's
Is
fine. That's fine.
I used to work in the Bahamas, and one of the things that we used to do was setting long lines to catch sharks. And these would be the ones that you had to check every couple of hours because we didn't want sharks being hooked up for very long. So it meant that we went out all day and all night going out to check them. And I remember I'd only just kind of started, and we went out in the middle of the night and it was pitch black. And I just remember turning around, and the whole wake behind the boat was all entirely lit up because of all the bioluminescence in the water. And it was just the most stunning thing to see. And it was quite, it wasn't unusual that that happened. Like, I got to experience it quite a lot. But I just remember being for kind of one of the first times I'd ever seen bioluminescence in the water, and actually to like just so much of it. And yeah, that was really, really cool. And it was a proper pinch me moment that I was like, wow, this is cool. This is what I get to do every day. So that one really kind of stood out for me. And it was a good day.
Yeah, it's so cool. Bioluminescence is just awesome. Like, there isn't any other way to describe it. Yeah, we had that. Especially when you're on a boat and the boat's moving and you kind of see it in these like waves across the on the side of the bow of the boat as well. It kind of feels like you're in some sort of like weird sci-fi film. It's so magical. Yeah.
Yeah,
It's really cool.
absolutely. It's yeah, it feels like kind of floating. It's like some kind of animated thing of how it would all be. It's really cool.
Yeah, like on a spaceship. But if we can kind of like go back to your childhood and growing up, I sort of wanted to explore sort of where your connection to the ocean came from and how you got to study marine science.
Absolutely. So, just as far as I remember, I've just always loved the water. I've always felt super comfortable in the water. I've always been really interested in the ocean. But I was also growing up just super obsessed with animals as well. I had pets growing up, and they were just my world, and everything about animals, wildlife, I just was super interested in. But when I was growing up, this is quite different to what any of my family members do or anything that I knew of anyone doing when I was growing up. So everyone when I was little kind of assumed I'd be a vet because I just loved animals.
Same.
And that was
Same.
what everybody, it was like, oh, you like animals? Yeah, you'll be a vet. There's no other options of working with animals. But I'm actually a bit squeamish, and I don't really actually like kind of blood and guts and things. So I knew that I would never make it as a vet. As a small child, I was like, I don't think it's for me. But I think I was about 10, and I went to an aquarium, and I ended up speaking to a member of staff, and they explained to me that they were a marine biologist. And I was like, wow, that's a thing. Like, I had absolutely no idea. I didn't even know, like, that a biologist was a thing, right? And so yeah, from literally that day, I was like, I'm going to be a marine biologist, which was a little bit ridiculous because I was a child and probably still didn't understand what it meant, but I was like, I'm going to do this. But when I was younger, I grew up in the Midlands in England, so I was a bit far from the sea. I used to go to the sea a lot because my family lived in South Wales, but my main way of accessing kind of animals and my interest in that area was mainly through documentaries. And I particularly remember watching a documentary when I was a teenager about the shark fin trade. And I
Oh,
was just
wow.
like, yeah, I didn't really know, this completely opened my eyes. And at the time, I don't think I really knew much about sharks either. And I remember watching it and I was like, oh my God, everyone hates these animals. Like, I felt like the perception of sharks, everyone hated them. And I was like, but they're really, really cool. And then I was learning how endangered a lot of them were and the shark fin trade issues. And I just immediately was like, wow, these are some of my new favorite animals. And I felt like I really had to fight their corner to a lot of people because I felt that no one liked them. So I was already super obsessed with everything ocean related, everything animals, and luckily science was my favorite subject at school as well. So it kind of all fit together. And yeah, getting to work with sharks down the line was actually just a bonus. The fact that I was always so interested in them growing up, that it kind of has all fallen into place. But when I went to university, I didn't actually do marine biology. I did zoology. So not too far off, but it was much more terrestrial based. But I still always knew that I wanted to do work in the marine environment. So I kept trying to do extra things and tailor it towards marine biology where I could.
Yeah. We had very similar experiences then, because I had the same thing. Everyone assumed I was going to be a vet or work in a zoo, either one. And then I didn't actually find out marine science was a thing until I was 17, and I was just about applying through UCAS, and my biology teacher was like, "You look like you're pretty interested in, coastal wildlife and the sea and stuff." And I was like, "Yeah, but don't want to work in an aquarium." And he was like, "Well, there is this thing called marine science." And it was just like, amazing.
Light bulb moment.
Yeah. And then the rest was history. But yeah, and the same thing with sharks too. I think my family were a bit worried about my reaction to like Jaws and stuff, because I was just like, this is the coolest animal I've ever seen. I think I'm maybe a couple of years older than you, because at the time there wasn't really any discussion around sort of the shark fin trade, or no documentaries about that. There were documentaries about shark attacks, and that was pretty much it. So yeah, awesome. And then that led you like all the way onto the path, like into your PhD. But you mentioned the Bahamas earlier, and I can't let you get away without talking about the legendary Bimini Biological Field Station, where you did a placement. And I was just wondering, like, how did that come about? And then, how did that influence sort of where you wanted to go in your scientific career?
Yeah, I was really, really fortunate that I got to spend some time at the Bimini Shark Lab. So how this came about was a little bit to do with when I chose to go to university. So I mentioned that I did zoology instead of marine biology. And I did this solely because that degree program allowed you to do a placement year. And at the time when I applied to university, it wasn't that common. There were a few places that did it, and they didn't offer marine biology, but they did offer marine placement. Not marine. They did offer placement years. So so that's kind of, it was the idea that I could get this work experience for a year that actually dictated which university I went to and what degree program I did. So it kind of started a few years prior when I applied to uni. I applied for a lot of positions, but Bimini was one of them, and I was just so excited when I got it. It was definitely my top choice, so I was pretty excited, especially to be able to work with sharks. This was really my first experience of getting to work with them in any kind of way and getting to see so many species and interact with them. There's just something I hadn't never had the experience to do so before. And it was a really amazing opportunity to really learn more about a career in research, actually, and what really research involved. And it was just a great experience all around and really opened my eyes to a lot of things. And the work that they were doing there was really interesting, and there was quite a few different research projects happening that I was involved in. But while I was there, I was learning also about the previous work that had been done. And there was a project done by Kevin Feldheim and others, and this was done a few years before I'd actually started. So it wasn't something I was involved in, but I was learning about it while I was there. And it's actually how they were using genetics to identify natal phylopatry in lemon sharks. So natal phylopatry is where an animal is born, they live their life, they come back to where they were born to give birth to their own pups. And they normally also come back to the same place every time they pup. So in the case of a lemon shark, it's every couple of years. So they were able to do this with the testing the genetics of these individuals to see it's the same individuals coming caught in the same location. They're testing, they can tell that it's their offspring, and siblings are found in these multiple years. So that I hadn't really done any molecular work, and I also say was not involved in this genetics project at all. But I remember being absolutely fascinated. I was like, this is the coolest work I've ever heard about in my life. And then when I came back to university, I was like, right, genetics is where it's at. This seems to be the coolest thing I've heard about the entire time I've been at university. And then luckily kind of dived into some molecular work in my final year of university. So even though I wasn't directly involved in it while I was at the Bimini Shark Lab, it was just one of these many things that was really opening my eyes to all of this amazing research that was happening, and kind of made me think that's something I really want to be a part of. So definitely defined where I went afterwards, and it was just such a great experience.
That's so cool. Yeah, because I mean, like that's the whole point of doing a placement, right? Is to figure out kind of what it is specifically that you're interested in. Because when you start university, you're so broad, and it's hard to know sort of where you want to go, especially as it's really helpful to have a placement like that where you're actually out in the field and experiencing what these techniques are going to be like, because that's so different to hearing about it in a lecture. But yeah, I'm so glad that was such a formative experience for you and sort of took you down the road of genetics and molecular biology. And of course, your PhD is now kind of focused on, you know, the tiniest, but arguably the most important parts of a shark, which is its DNA. And I wondered if we could talk a little bit, for someone who is, you know, pretty new to the field, what sort of things can we learn by looking at a shark's DNA?
Yeah, absolutely. So there's so many different things that we can address actually with using a DNA. So it's just to start off, I like to think of DNA as the molecular blueprint of how every organism is, what it looks like, how its body works. It can influence, it influences and is the cause of everything about you. So it holds so much information. And if we can tap into that information, we can learn a lot about these individuals. So just a few different examples that I'd like to highlight as to what we could do, and this is by no means an exhaustive list. So
yeah, I'm asking you to talk about like a very huge field with
yeah,
many different factors, but
so just a couple of examples.
many different avenues.
Yeah. So, and what I also would like to highlight is actually we can use DNA to not only learn about a single individual, we can learn about a population, so maybe a group of individuals over a certain area. We can learn about a species, or groups of species, and we can also learn about the ecosystem in general. So on an individual level, we can identify what the kind of species is. So particularly for sharks, this is really useful in terms of trade, because you might get a body part. We can't identify what this animal is by looking at it, but using its DNA, we can tell you exactly what species it is. So that can be really useful. We can actually also learn about the individual's health. So we can actually research into its immune genes. So again, this is all sorts of information we could find about the individual itself. On a population level, particularly for sharks, we can use this to classify stocks. So we can know if species in different areas, well, the same individuals of the same species in different areas, whether they are mixing with each other or whether they're completely closed off from each other. And this could be by distance, or this could be because of a physical barrier. There's all sorts of reasons, but we can use DNA to find out, are these animals mixing, and do we need to manage them separately, or can we manage them as one group? But also for a species level, we have a lot of animals that actually look the same. So we can actually use DNA to figure out, are these different species? And we can classify groups of animals. Obviously, traditionally, this was done by looking at them and taking measurements and describing features, whereas now we still do that, and that's such important work, but we can also add in the genetic element to identify, and quite a lot of species over the years have been reclassified or changed or split up because of looking at their genetics. We can also look at this on an evolutionary timescale as well. So we can use genetics to see how animals have adapted, and kind of think about the evolutionary timescale and what has changed over this period. So yeah, DNA also helps with that. But as I mentioned, we can kind of get information on ecosystem. This is more to do with, an example here would be for diet. So not only can we look at the DNA of a particular individual, we can actually look at the DNA of the individual animals that it's eating. So we could take stomach or fecal samples, and we can actually we can get the DNA of those animals as well, and either for species identification to know what they were eating. So we can delve into that in quite a lot more detail as well. And that's actually where I started when I first started molecular work. I actually was doing diet analysis of breeding European storm petrels. So did birds for a short while. Not hasn't all been sharks. Um,
Love a storm petrel.
yeah, they're really, really awesome. Not biased or anything, but definitely my favorite bird. And
They're amazing. For people that are listening, sorry, just like really, really quickly, they're like this. They're this incredible seabird that travel, migrate huge distances, travel in, really pretty gnarly conditions as well. Like I've always seen them in high winds, high waves, but they're tiny. They're not very big birds at all. Yeah, they're so cool.
they're such tiny birds. They'll sit in the palm of your hand. They have gorgeous little faces and make the coolest noises. If anyone gets the chance to maybe look it up on YouTube, the sound that they make is really, really bizarre, really, really unusual. And yeah, so a very, very cool species. It was great to do some seabird work for a while. But that's also just to highlight all of these methods that are molecular methods or DNA-based methods. They're not just for sharks, they're literally for all animals, or not even animals, you can also use it for plants or fungi, other living things. Like all of these techniques can kind of be applied for everything. So it's not just a field or these questions you can't just ask for certain things. It can be applied across all living things on various scales. So it's pretty cool.
Yeah, absolutely. There's so much that we can learn. It just boggles my mind that this thing that we can't even see with the human eye can tell us, so much about an animal or a plant or, a fungi's life and how they operate and move around the world and how they used to be. And it's kind of like unlocking a secret code or, a secret set of novels or something that tell you about that animal's life. It's so, so cool. Speaking of cool things, we're now going to move on to your PhD research that you're currently completing, and your research looks at population genetics. So if we think about, what we've just talked about, DNA, the field of molecular biology, massive field with lots and lots of different disciplines within it, and one of those is population genetics. And just before we kind of get into your specific what you're doing specifically for your PhD, I wondered if we could just quickly explain sort of what population genetics, what does that actually mean?
Yeah, of course. Yeah. So population genetics mainly revolves around questions to do with, are individuals found in different places connected to each other genetically, or isolated from each other, or what we also call differentiated, so you can tell them apart using their genetic information. So what we often want to know is, are they mating and are they mixing these populations between areas? Obviously, with a lot of shark species, not all, but a lot, they do move quite a lot. We have some that are quite resident, but generally we have ones that move around a lot. So for a mobile species, just by looking at them and catching them in certain areas can't really give us information as to where the kind of boundaries of these groups are, if that makes sense. So tagging information can show us how far they're moving, but a species might move, but it might not actually mate with individuals in another location, which means that genetic information isn't being shared and isn't being passed. So this is why we kind of want to look at these population level genetics, whereas quite a few of the earlier examples we were talking about were kind of, are they present in an area, what are they eating, that kind of thing. This is now for the species, where are the boundaries of these groups within the species. Hopefully that makes sense. So you get these lack of connection generally in the natural world due to physical geographic features which stop them. So if we think about terrestrial animals, the land animals, if they're on an island, you might have the same animal on multiple islands, but they physically can't access each other, so they can't mate and we don't get any genetic mixing, even though they are the same species. It's a little bit less clear in the marine environment, because, as I say, especially for a lot of species, there aren't really these geographical boundaries, because there's less. There are some forms of boundaries in the ocean, but less compared to when we think about the terrestrial environment. One of the other ways that we can also have a lack of mixing and a lack of connection between populations is also by distance. So, for example, if you take the Atlantic Ocean, animals on the East Atlantic might be genetically different to those on the West Atlantic, but then the ones in the middle of the Atlantic might be kind of look similar in ways to both of them. And that could just be to where migration limits go. So they might travel so far, mix with them somewhere else, and then but they don't get further. So yeah, genetics allows us to investigate this a little bit more. We can see which ones are more closely similar to each other or not. So that's kind of, we can ask some more detailed questions within that, but that's generally the rough concept of population level genetics and why we might want to look into those things.
Yeah, that makes perfect sense. You explained that beautifully. So you're looking at those kind of questions for your PhD to do with the spurdog, aka the spiny dogfish. And just before we kind of go into like the population genetics of the spurdog and sort of what it is exactly that you do, I wondered if we could spend a little bit of time shedding some light on this species, because they don't often get talked about all that much, but they are a pretty special species. Let's start off with kind of like what they look like.
Yeah, absolutely. So spurdog is actually quite a small species of shark, and we get them pretty much all across the world. They like the kind of more cooler temperatures, so we get them more in the temperate areas. We don't see them in the tropical seas. They can grow to about 2 meters in length, but generally we don't really see them much bigger than 1 meter. I think that is the maximum. But yeah, they're generally about 1.2 would probably be considered a larger-ish spurdog. And yeah, like most sharks, they're very long-lived, they're slow-growing, and they don't have that many young compared to bony fish. And one of my actual favorite facts about them, which really surprised me when I started working with them, is they actually have a really long gestation period. So they're pregnant for up to about 2 years. And this is potentially similar to or longer than an elephant. And these animals are small, right? Compared to an elephant. You can understand why an elephant takes that long to make a baby. And this just completely blew me away when I started working with them. So they're said to have one of the longest gestation periods of any vertebrate out there. So
poor female spurdogs.
Yes, I know, bless them. And so when they have gone through this really long gestation period, they can have anywhere between 1 to 32 pups at a time. And for those of you that might be a little bit more familiar with sharks and their weird ways, these animals are what we call ovoviviparous, which means that embryos grow inside the uterus and then nourished by an egg instead of a placenta, and then they will have a live birth. So yeah, I won't go into shark reproduction too much, but
it's okay. If you want to go into shark reproduction, go back to episode 1 of the podcast. There we go, and we talk about that. So yeah,
one of my favorite things about sharks, I'm like, they're so different. Like there's so much variation. Yeah, so they're really cool. But also to kind of see them and identify them is they actually have venomous spines on the front of both of their dorsal fins as well. So this is where they get their name spiny dogfish from. And it's one of the only venomous marine animals that we have in the UK. So pretty cool.
Pretty badass little shark.
Yeah, completely. But one of the things I wanted to kind of highlight to people is, even though, spurdog don't get spoken about very much, I feel like they're kind of underrated, particularly maybe in the shark world, because they're not these big, impressive animals. But actually, a lot of people probably come across them because they traditionally were used for most of our fish and chips back in the day. Probably also not unheard of now that you might come across and eat them, but not so much as it used to be. So they were really, really common. They were commercially fished for a long time.
Yeah. But are they still quite largely caught as bycatch?
Yeah, so there was a prohibition on retaining spurdog since 2010 in the Northeast Atlantic, so UK and EU. But actually this year they've opened the fishery again. So yeah, so up until 2010 they were caught heavily as commercial species. Since 2010, you couldn't keep them. But the challenging thing with spurdog is if you encounter them, there's not much way to avoid them, particularly with large nets. So trawl nets is one of the most common ways that they're caught. These are nets that are dragged along the seafloor, and they're non-species specific when they're catching fish. And so if there's spurdog in the area, you will catch spurdog. There's sadly not much we can do about that. So bycatch was still an issue even when it wasn't targeted, or even now, even though there is a small quota for them, fisheries, they might not want to catch them because that's not what they're targeting that day, but they will still come across it, so it can still be thought of as a bycatch species. So they actually get quite a bad rep with a lot of fishers because they're quite annoying to handle, especially with their spines. You want to avoid them, and when you have quite a lot of them, because you can catch quite a lot of them in one go, and you're trying to empty them all out of your net, but then you've had to spend all of that time on a species that you can't keep and you can't sell, so you're discarding it dead or alive. So a lot of people find it quite wasteful. And then you haven't been able to capture the species that you actually wanted to catch. So they tend to get quite a bad rep in the fishing world as well, because people find them such a nuisance. So targeted or not, fisheries can still be quite an issue for them.
Yeah, and what's their conservation status at the moment?
So according to the IUCN Red List, they're vulnerable worldwide, but they are classed as endangered in the Northeast Atlantic. So the stock in the Northeast Atlantic is definitely doing worse compared to the rest of the world. Yeah, and they had quite significantly declined since the 1970s, likely due to increased fishing pressure and the industrialization of the fishing fleets.
Yeah. And of course, like you said, the sort of methods of fishing is a factor in why so many of them are caught, but also linking back to your research, one of their traits is that they tend to aggregate. And that's sort of one aspect that you're researching with your PhD. So can you tell us a little bit more about this? Like, do we know why they aggregate and how big are those aggregations?
So the aggregating nature has kind of been part of their decline, I feel, because when these animals aggregate, they can aggregate in extremely large numbers, and we're talking thousands of individuals come together. So this is when it's unfortunate, and as I say, it's very difficult for a fisher to avoid them if they're in the area, because if they pass the net through, there are many spurdog, and they'll just get an entire net full of spurdog. So if you can't sell any of those individuals, that's very, very inconvenient for your day. But yeah, and with spurdog, because they were actually a commercially important species, they're actually one of the most well-studied elasmobranchs compared to many others across the globe. But we still actually have very limited information on their aggregating behaviour in terms of the drivers and why they are together. And also, in combination, in terms of leading to my work, is we don't have that much genetic information for them either. So I, for my PhD and for my Save Our Seas funded project, I'm looking at these spurdog aggregations, and I want to know a little bit more about the genetic composition of these aggregations. And the reason why I'm looking at that, because I guess to start off with, when we think about sharks that group together, there are many other species of shark that school and aggregate. And if you're thinking about a mobile shark and they form these aggregations, I would assume that these animals are probably not related to each other. But some previous work by James Thorburn and others, also at the University of Aberdeen, he was also studying spurdog and was looking at the population genetics. And he had some samples from a single catch, and his data indicated that there might be some full siblings. So this wasn't common across the other groups that he sampled, but there was just one group that potentially had full siblings. So that was really kind of unusual and unexpected. But at the time, and the methodology that was used, we couldn't say which individuals might be related to each other, and we couldn't say really, it came up as full siblings, but we couldn't really trust that it might be an actual true full sibling relationship, and we don't know which ones were. It was just kind of how the analysis presented was like a proportion of this group is related, is kind of what would come out. But genetic techniques advance very, very rapidly. So even in a few years, what we can do with this information changes quite dramatically. And that's kind of where I came in, is we have better techniques now, and we kind of had like a little hint to there might be relatives between spurdogs that were caught together, but we didn't really know any more, and we kind of still had a lot of questions relating to this. So I wanted to delve into this further. A few years ago in 2015, there was a rare event which has also enabled my PhD work to happen. So off the coast of the Isles of Scilly, so this is down on the southwest of the UK, there was a bycatch event, a spurdog, in very large numbers. So this was estimated to be a 10-ton catch in a single net.
Wow. Okay.
And yeah,
that's a lot.
It's a lot. So you can see the scale of that happens when you get these aggregating animals. There are so many together in one go, and you have the potential to remove so many in just one net. But for those poor boats, they weren't they didn't want to get any spurdog that day, and they had so many that they physically couldn't move the net, so they couldn't release the net, they couldn't release any of the animals out because it was so big they couldn't pull it onto the boat. They actually couldn't even move the boat, so they were kind of stuck, and they had to get towed back to shore. And because they were towed back with the whole net full of all of these sharks, they were landed. And this was at the time where you weren't allowed to land these animals, but they were due to the extenuating circumstances that they found themselves in, this was allowed. And all of these sharks were actually collected by CFAS, which is the Centre for Environment, Fisheries and Aquaculture Science here in the UK, and collected for scientific purposes instead. So this became a really unusual opportunity to study an aggregation, because I think one of the reasons we are limited in our aggregation knowledge is we don't have access to a whole aggregation, to actually find out information about them. And then this just random event has allowed so many scientists to gain access to these samples. So we did a mass dissection of these animals, and literally every single part of this animal was taken to somewhere around the world to a different research group to learn more about spurdog. And for me, I was able to take a tissue sample from all of them, which is what we need for looking at our DNA. So we had all of these individuals that we know were caught in the same sampling event. So this has kind of given us a chance to look into the genetic composition of these animals on a larger scale than has ever been done before, because for previous work, you kind of might get like 10, if you're lucky, maybe 20 samples from a single catch, and then we'd have multiple of those and we'd compare them to each other in our population genetic level research. But I actually was able to obtain 656 samples, and this was only estimated to be 1/5 of the catch. So I don't have samples from every individual that was caught, and obviously in terms of the aggregation, there are probably individuals that didn't end up in the net. There might have been individuals a part of that aggregation that weren't caught. So we don't have every single individual, but we have a pretty good sample size compared to most genetic research, when we normally deal with a few tens. At most.
I would say that's a huge. That must have taken a long time to analyze as well.
Yeah, so I will say I haven't actually been able to analyze all 650
That's understandable.
Samples.
That's understandable, yeah.
But they are there, and it's something that we may get to be able to do in the future and complete the analyses for all of them. So one of the aspects I wanted to look at is relatedness. So the fact that James's work suggested, that there could be related individuals that are together. So this is one of the main questions I had for this work and to delve into with these samples from the aggregation. And one of the reasons why I want to know this is because if there were related individuals in these really large groups, does that mean that these groups as a whole, or maybe smaller groups that form a large group, could have actually been together their whole lives? I think a lot of people, I think the assumption was that these groups form. So maybe, we don't know why, maybe around feeding, maybe around mating, maybe due to environmental conditions. But at least my understanding is a lot of people think that it's not particularly long lasting. I don't know what scale we're talking on, to be honest, but the idea that you could find siblings, does that mean that these siblings have stayed together their entire life? And then we're catching them in these aggregations. So if we didn't find siblings, then that's probably the assumption that we would have expected. If we would find siblings, maybe it would mean that this could indicate that there's something a bit more to these groups, and they might be a bit more long lasting. Because if you would expect maybe relatives if you were testing individuals at maybe a nursery site, or for a highly resident species, that wouldn't be, it's not like you can't find related individuals for sharks, but it's just these kind of more mobile species that's a little bit more surprising. So that's kind of how the project came about. It was, yeah, it kind of really fell with the timing of just the samples that were available, and that's kind of why we wanted to look into this. But I can tell you that for the part of, I'm not finished entirely on this project, but so far I have actually found quite a lot of relatives, and we found a combination of half siblings, first cousins, and half first cousins. So there's a range and a variety of levels of relatedness, and they're also not just pairs. So we don't just get a half sibling here, a half sibling pair there, a half sibling pair there. We're actually getting like little family clusters. So I have individuals, for example, I have one individual that's related to 12 individuals in that aggregation. So we're definitely seeing some clustering, more than I was expecting. So it's been quite cool. I'm still delving into the analyses in more detail and trying to, piece together quite what this might infer for the group. But as a first step, it's looking quite exciting, or at least I think it is.
That's really exciting. No, that is really exciting. Like, I love it when you get surprises from science, because that's what it's all about, right? You've just got a question, you don't really know what the answer is going to be. And this is kind of like a curveball question, so feel free not to answer it. But what if we are finding related individuals hanging out together? Is there like any advantage of the animals doing that?
I haven't actually thought about it that way. That's a really good question. I'm not entirely sure evolutionary if there'd be that much advantage. I'm not entirely sure if they're even like actively choosing to be with a relative. My theory, so by no means is this is proven at all. My current thoughts are that they're probably, spend some time in a kind of nursery area, pupping area. Those that when they get to a certain size, they're then probably leaving that area. And those that are a similar size to them are potentially likely their siblings. And I think it's then those that have been kind of spend their early years together, similar kind of size together, they're then continuing to be together. And I think then it's just chance that they're siblings. So they could be spending their life with similar sized animals from the same nursery site that happen to not be related to them, but you also might be getting ones that are. And also if there is natal phylopatry for spurdog, that would explain that for example, you'd have a mother giving birth to the pups, but then her siblings would also be going to the same place to pup. which means that one offsprings would be near the other one's cousins and things like that. So this is where my mind is kind of going with what the possible explanation for this occurring. But yeah, there's still a lot of unknowns. Classic with science is you have all these questions, and the answers to your questions provide more questions. Yeah. So you're getting a tiny bit of a piece of the puzzle, and then you get that little nugget of information, and then you're like, oh, well, there's now 10 other questions that we have to try and understand this better. So,
More questions. Yeah.
yeah, it's never end.
That's what I love about science, though. I think, especially because of the time that we're in, and the sort of there's that need to find the answers to these questions, sometimes relatively quickly now. But that's how science started in the beginning. It's just kind of like, I wonder why this does what it does, and then kind of like branches out from there. But I think that's really cool, and like a really, really exciting finding potentially with your thesis, which is very cool. I very much look forward to seeing kind of like what comes out of that. I just wanted to, just before we kind of bring the episode to a close, I wanted to spend a little bit of time sort of talking about like the conservation implications, because we've talked a lot about genetics in this episode, and particularly concepts like connectivity and diversity and relatedness, and I just wondered, we kind of touched on it a little bit, but why is this important to understand from like a conservation perspective?
So one of the reasons we want to understand the extent of related individuals within aggregations, because if we had whole family groups traveling together, which could also be caught together, these family groups could have what we call adaptive traits. And if they're caught, that adaptive trait won't get a chance to integrate into the wider population. So the issues with removing families is more of a kind of on an adaptation and kind of evolutionary impact, because it reduces the chance for these selective adaptations, which might be really beneficial for a species, to be removed. Because you end up starting with, if one person's got a good genetic mutation, which gives a good adaptive trait, you're going to pass that on to your offspring, right? And so when this kind of mutation first comes about, initially you're finding it in these related family groups, and it takes a lot of generations for it to mix wider into the population. So if you are removing a family group, if they have any of these kind of good mutations, you're not allowing that to be passed on to elsewhere. So the issues that could come from having relatives in groups are generally on a kind of adaptation scale, but in addition to obviously removing the number of individuals from the population. But as I say, they have kind of opened the fishery now to a limited extent, so it has indicated that the population has been recovering, and that could be possible. But why we want to know about the genetics in a bit more broader sense, in the connectivity, is, as kind of mentioned before, particularly for sharks, the kind of main application of that is understanding stocks. And if we understand the stock, we can then apply the best management and conservation practices to that. So for example, if we have two different stocks, say stock A and stock B, if stock A is depleted, so there's less individuals, and we have a healthy stock in stock B, we need to know if they're mixing, because if they're not mixing, stock B can't help stock A keep its numbers up, and it can't pass on its kind of genetics, and it won't have any mixing to increase the numbers in that area. So that has pretty big conservation implications, because if we treated them as one group, the conservation measures, they're going to need different conservation and management if we have one really healthy, high population stock and one low population stock. Because if it was one group, you would assume that the healthy lot could help the ones with not many numbers left, and that might not be the case. So yeah, particularly for fisheries, which is one of the most applicable kind of managements for shark, this is quite key to understand. We can also find, mentioned before when we were talking earlier about identifying different species, with the genetics. And actually for spurdog, there is a subspecies called the North Pacific spiny dogfish, which is found in the North Pacific. And it was actually through looking at the genetics, it was deemed a different species in 2010 compared to all the other spurdog around the rest of the world. So again, that is quite a big thing for management, that you need to know that actually this is a different species, and actually they might have different life history traits and they might be affected differently. So it's not only about identifying the stocks, it's identifying, are these actually the same species or not across the world.
Yeah, absolutely. And thinking about just going back to your adaptive traits just real quick. So it's kind of like thinking of how to describe it. It's like if you had, so say we've got climate change, you've generally got global warming, and so you might have groups of families who have a trait that allows them to deal better with warmer conditions, and if you are taking them out of that population, then that adaptive trait doesn't get the chance to spread, and so you then are left with a population that potentially can't adapt to warming seas, if that makes sense. So that's kind of like the longer term connotations. I don't know if I described that.
Yeah, no, that is absolutely perfectly described in terms of, yeah, as in a hypothetical situation, that is what we're looking at.
Yeah. Oh man, it's so fascinating, and there's so many kind of like little tangents that I could take us on and go down, but I'll not do that. But it has been an absolute pleasure to sort of dive into the world of, genetics, DNA, spur dogs with you, and I'm sure all the listeners will be the same as me in being really excited and sort of following where your work goes. And with that in mind, what comes next for you? I imagine probably a lot of writing.
Yeah.
And also, where can people follow your work?
So I have a year left of my PhD, so I'm going to have a very busy year ahead of me. And the work that I've specifically spoken about in this podcast, I'm actually wrapping up now, and I do actually hope to submit to publish it later this year. But Will, as you say, I'm going to have a busy year doing lots of writing, lots of finishing up on my other projects. But in terms of following the work, you can find me on Twitter. I'm at fenella.wood. And so, yeah, when publications come out and anything particularly exciting, I'm sure I'll be posting about on there. But you can also find out more about the project on the Save Our Seas Foundation website as well, particularly about the work that we've spoken about today. And then beyond the PhD, I am not sure yet, but I'm hoping would be amazing if it could still be something molecular, and even better if it could be continuing the work to help sharks. But we shall see.
And a holiday.
What it brings. Oh yeah, a holiday. That's definitely very high on the cards. Yeah.
A nice rest.
Yes. So yeah, most people's plans post PhD, it's always about where you're going on holiday afterwards tends to be the biggest discussion point.
Yeah, I went straight into a job, and I would not recommend doing that. I'm always like, please make sure that you take your mental break. But yeah, but that's fantastic. We'll link to everything in the show notes, as we always do, and you'll be able to find all the details on how to find out more about Fenella's project, and also Fenella herself down there. So make sure that you do that. And finally, we do have one more question, if you'll humour us for just another minute. And that is, and it doesn't have to be a spur dog. A lot of people think when I ask this question that they have to be their study species. You don't. But if you could be any species of shark, ray or skate in the world, what would you be and why?
So I think you're going to like the answer to this one, Isla, because I would like to be a basking shark.
Yeah.
And well, it's one of my favourite species of sharks, so that's always a win. So I do agree with you on that front. But they just seem very chilled out, and they eat a lot, and they hang out in groups, and they just seem to I just love them. And then they're just so surprising, incredible when they're like kind of slow and chilled, and then they're just like breach. And you're like, well, I didn't know you had that in you. So I just think they're such cool animals. So yeah, if I was going to be a shark, I would like to be as amazing as a basking shark is.
Oh, you've made my day with that answer. I love representation for basking sharks. And yeah, they seem to live a very lovely and a very placid existence. No, that was a great answer. And with that, I will bring it to a close. But Fenella, thank you so much for taking the time to come on the podcast and chat all about your super fascinating research. It's been an absolute pleasure to talk to you.
You're welcome. Thank you so much for having me.
The World of Sharks podcast is brought to you by the Save Our Seas Foundation. It was hosted and edited by me, Isla Hodgson. Our beautiful artwork is by Jamie Silva, and the wonderful jingle you can hear right now is by David Knight. An enormous thank you to Fenella for coming on the podcast and teaching us all about your work and the wonderful world of the spurdog. You can follow her over on Twitter. She is at Fenella_Wood. And thank you at home for listening. If you like this episode, it would be awesome if you could just drop us a rating and a review on your podcast app. We love hearing from you. It helps us improve and helps us spread the word about how amazing sharks are, and who doesn't want that? If you have any topics you want covered, or you just want to say hi, you can also get in touch by emailing isla@saveourseas.com. And you can find out more about the Save Our Seas Foundation and the work that we do by heading to www.saveourseas.com or following us on social media. We are at Save Our Seas on Twitter and at Save Our Seas Foundation on Instagram. Alrighty, have a jawsome week, and we will see you next time.